Hello class and welcome back. My name is Ken Vitale. I'm a university professor teaching anatomy and physiology here. In this series, we review some tips and tricks and some extra learning points, some mnemonics. We're up to the renal system this week, and for the picture of the day, living in the Southwest, we got a lot of deserts, and you got to think about your renal system, at least I do. Getting dehydrated, you got to protect your kidney function, so I wanted to put some kind of Southwest vibe, and I thought this was a pretty good job. Okay, let's get going. You better love your kidneys because, when you're in love, right? Here's another drawing that's kind of a picture frame of a kidney with some kind of impressionism and a little bit of abstract art there.
Okay, so just like we covered in lecture, the urinary system is considered part of overall excretory anatomy, and some students are having some trouble with remembering the difference between internal and external anal sphincter when it comes to the rectum, or quote unquote unfinished business at the end when we get to the final stages of going through gastrointestinal system. So one way, just remember there are two rings in terms of the internal and external anal sphincter. Get my, there we go, pointers here. Two rings, two controllers, two different functions. So internal anal sphincter, just think of the eye as involuntary. Innermost of the two rings, it's smooth muscle, which is autonomic or automatic. And, well, basically this is under involuntary control via the autonomic nervous system. This sphincter blends with the, internal gut tissue and the intestinal wall there. This is not part of the external system, which we're going to cover in a minute. So hopefully that's one way to remember. The external anal sphincter, just think of, E for effort, because when you strain to go to the bathroom, you got to push hard, and sometimes you get an E for effort when you are able to, release the bowels. This is the outermost ring, and this is skeletal muscle. So skeletal equals striated muscle. So think of just like regular muscle when you're lifting weights, you build that for strength. Your external anal sphincter is under skeletal, voluntary, somatic control, self-control. This is the sphincter that we contract when we have to hold it in until there's an appropriate time. This sphincter, the external one, blends with the pelvic floor muscles. And a lot of people, when they think of pelvic floor, they think of the Kegel exercises. Women use that to build the pelvic floor muscles during pregnancy. And, men can do that too. I don't know if you've ever tried contracting your, pelvic floor muscles, but when you are doing that, you are also contracting your external anal sphincter. So maybe this is a little bit weird, but if you're thinking about, we're continuing to be gross here, I'm in automatically, right? Your stuff is in there, but to exit takes effort. So the internal anal sphincter holds things in, and then the external one, when you are ready to release, takes the effort. So the I and the E, hopefully that maybe helps you remember. Please don't push and bear down. That's another way to get hemorrhoids. Remember from the prior lecture there. Unless you sit with proper anatomy, now that you can use your anatomy to your advantage here, this is why some people have those squatty potties and things like that, where they push their knees higher up to align their external anal sphincter and their pelvic floor to release the bowels a lot more effectively. So if you don't do that and you push and strain, you can increase venous pressure, and that's another way to get hemorrhoids. And that's from the last lecture. Okay, on to the kidneys. There's a famous line from one of our anatomists: if you can draw a kidney, you can draw a moose. I'm terrible at drawing, but I guess I can draw a moose now. So let's talk about renal blood flow. This can be a little bit difficult, going from afferent to the glomeruli, efferent to the peritubular capillaries, vasa recta, and onto the veins. But this is a very important pathway to remember. The glomeruli, by the way, are also incredibly critical to our body. They may look a little funny when they're drawn like that, but this is literally what they look like. It's an electron microscope there. And this contains a ton of blood flow. About 25% of all cardiac output goes right through the kidneys. And another famous line, not from an anatomist, but from a nephrologist back at the med school where I went to: function of the heart is to pump blood to the kidneys. It's kind of a matter-of-fact sentence, but the nephrologist was trying to emphasize just how important the kidneys are, since they attract or take on so much blood flow through the body that all the heart does is just pump blood to the kidney. The heart's not important. Your kidneys are the most important part of your body, at least according to the nephrologist. Okay, one way to remember this, if you think of the glomerulus as a coffee filter, your blood is the coffee grounds and water, and the filter, or the glomerulus, only lets the brew through, right? The filtrate coming down through, and let's get my pointer, there we go. Coming through and across the glomerulus, it's only allowing the good stuff, the coffee, through. So the coffee grounds, which are proteins and cells and other things that you do not want to allow through, stay behind. Of course, we don't drink urea. That would be disgusting. We drink coffee, but I hope you get the idea. Sorry, that maybe this is not such a good visual metaphor, perhaps, but the brew goes down that nephron and becomes modified till the urea eventually becomes urine. Okay, maybe that was gross, but hopefully it's a way to help remember. Here's another quick review in terms of pathway of that urea filtrate through the nephron. We have the glomerulus up here, the PCT down and up the loop of Henle into the DCT, and then finally into the collecting duct. These are a lot of steps. Let's just go right through them before we get to the mnemonics so you know what you're dealing with in terms of all these letters. The mnemonic here, if this works for you, go buy pretty lacy dresses because, can't miss really unique and beautiful underwear, because this is the urinary system after all. So of course we'd have underwear in a mnemonic. Glomerulus and all the way down. Maybe this is a good way to recall what's going on. This is more microscopic anatomy down to the collecting duct, and then we have macroscopic or gross anatomy when we get into the calyces, the pelvis, the ureter, and then the bladder. There is another mnemonic out there that I don't like as much. It's kind of weird. It's kind of the direct anatomy mnemonic, but hey, if it works for you, go with it. If you have a better mnemonic, please let me know. I love to add students' mnemonics into these videos. I think they're great. This is how we all help each other learn. Some students also reach out and say, hey, Professor, I'm getting a little confused between cortex and medulla. What's in what? I went through things pretty quickly in lecture, and sorry if that was a little confusing. We don't really emphasize too much of that, since a lot of this is delving more into the world of physiology, but let's clear that up a little bit. So, cortex, pretty straightforward here. We have the glomerulus, and then the proximal convoluted tubule, or the PCT, then the distal convoluted tubule, and then finally the peritubular capillaries, which are surrounding the PCT and the DCT. And that's really it. Medulla is where things could get a little confusing. The loop of Henle does start in the cortex, but it mainly extends into the medulla, that descending and ascending limbs. The collecting ducts also run into and through the medulla. They got to come from somewhere and go somewhere, so sometimes people might misinterpret collecting ducts as being a cortical structure, but it's really categorized as a medullary structure. The vasa recta hang out with the collecting ducts, around the loop of Henle, and help maintain osmotic gradient. And then finally, the renal columns. That's another area that switches from microscopic to macroscopic. Between these renal pyramids, we have renal columns, which are, yes, indeed, extensions of cortical tissue that separate those pyramids. So this might be a little confusing. I get it. Certain cortical structures have to run through the medulla, and you might get confused in terms of where to categorize them. But PCT and DCT are cortex, loop of Henle and collecting ducts are medulla. Peritubular capillaries are associated with the cortex and cortical structures, while vasa recta are associated with medullary structures. Hopefully these drawings also help kind of illustrate where we have the cortex and then the division and then the medulla down there. Here is where the columns come through. The renal columns extend between, separating those pyramids. And then this is a nice thing where we have microscopic structures overlaid onto macroscopic gross anatomy. And hopefully this helps pull it all together and gets rid of the confusion. And now for the last part of this video, we are going to go through histology. And I get it, this is very difficult and it just looks like a big sea of pink and purple, swirlies all over the place. It is hard, unless you know what you're looking for. So I want to arm you, get it, anatomy joke? Well, that was terrible. Some distinguishing features, some mnemonics, and maybe some pattern cues through that pink and purple mess. And then maybe some visual metaphors that might be questionable. Sorry, if you have better metaphors, please let me know. I am all ears. Another anatomy joke. Okay, so let's start with the glomerulus. So you can think of tuft in a cup or ball in a bubble. There are different ways to kind of say that, but you're basically looking for a nice round ball of capillaries, histologically speaking, in a little white circle or capsular space surrounded by basically simple squamous epithelium. And another way, instead of the ball in a bubble or tuft in a cup, a sun in a bubble. And yeah, when you kind of visualize that, I guess that works. This is a nice little ball of capillaries here and surrounded by this, hang on, get my pointer here. There it goes. This little white space right there, this is your Bowman's capsule. On to the PCT. Fuzzy with friends, maybe. The PCT are the most numerous in the cortex. They have a fuzzy lumen. There's a brush border of microvilli, and the cytoplasm is a little more pink rather than purple, if that helps you. But the easiest thing, if you just look for the fuzziness, that's where you're going to find it. Since there's some stuff in microvilli in that lumen, the lumen often appears like it's just occluded or like a messy, dense little mess because there's a lot of absorption going on. And when you start looking at these in cross-section here, these tubes, then you have this one that's got all this fuzziness and mess in there. Yeah, that's your PCT. Contrast that to the DCT right there, which we're going to get to next. So here's your fuzziness, a lot of friends around there. Sorry, glomerulus is over here, the distal convoluted tubule there, your proximal is right there. So fuzzy with friends. All right, on to distal, clear and compact, right? This is what we were just alluding to before. Less numerous than the proximal, no brush border. There's a clear lumen, and it's just this nice round circle that the cells are more defined, also with pale cytoplasm. But this one, to me, just looks like circular or round donuts, right? I mean, to me, that looks like donuts. There's a lot of food references in anatomy. It just is what it is. But that's your distal convoluted tubule. Some people say, wait, that kind of looks like a collecting duct, or maybe that looks like a loop of Henle, and I get that, but think of your fuzzy with friends. Your distal is going to be near your proximal, and that's also going to be near your glomerulus. So if you see a lumen here that is wide open, that's probably a distal, not what's in the medulla, which we're getting to next. If you zoom in a lot farther here, this star showing some little bit of fuzziness there, that is a proximal. Right next to it, right there, that donut, I don't know if that's like a jelly-filled or whatnot, that's your distal. Over here, this star is showing a distal. Right here, there is a proximal. Hopefully that helps. Here's a glomerulus right there, and this is part of the glomerulus there, that sun in a bubble. All right, on to the medulla. We're shifting. There is a thin and thick segment that's kind of hard for the purposes of our class. We just want you to identify the loop itself. You don't have to say if it's the thin or the thicker one. But these are very thin-walled. They're very simple squamous epithelium. They kind of look like capillaries if you're used to looking at capillaries in histology. But they have no brush border. Just look for these thin-walled structures, and because there are loops oftentimes in the section, you're not going to get it in cross-section like a little circle, like a donut. You're going to get them longitudinally oriented right next to each other. And FYI, there's going to be no glomeruli because we're in the medulla. So that's this slide here. We have these little loops right here, very thin-walled right there. Contrast that to this one, which we're going to get to in a moment, very thick-walled cuboidal cells right there. So that is your loop of Henle. Here's another one here, thin-walled right there. There's a bunch of them near each other. You could argue that that's a loop of Henle right there, as opposed to collecting duct, which, sorry, this is CT instead of CD, but it's the same structure. Let's get to that. Collecting duct right now. These are cuboidal columns in a row, I guess. That's about as best I can do. It's not really a good mnemonic for that one. But these are just standing out, so to me at least, so clearly. Very square cuboidal cells, very distinct cell borders, and very large, wide open lumen. These are, well, to me, they kind of look square like tiles that you're tiling up and down, and they're very well organized and clean. So here we go. This is a loop of Henle, very thin-walled, if you can see that cell wall right there, and contrast that to that car tire, thick, cuboidal structured collecting duct right there. That is, to me, these are all standing out as collecting ducts all along here. That's a loop of Henle there. Loop of Henle, loop of Henle, loop of Henle, collecting duct right there. So I hope that helps. A mnemonic to wrap it all up. Good people do laundry cleanly. I'm sure there's a better one that you can come up with. Please let me know. Send it via comments or whatnot. I would love to include it in a video. But good people do laundry cleanly. Glomerulus down to the collecting duct. And then here are the buzzwords there, right? The ball and the bubble, the fuzzy filled lumen, the clean donuts, the thin-walled loop, and then the big lumen, thick cuboidal collecting duct. So hopefully that helps. I'm going to leave you with this one. The nephron, I guess you could think of it as your spa day. Probably be the weirdest spa day ever here. You enter the bubble bath, right? The glomerulus going through, right? Ball in a bubble. And then you got to exfoliate with that fuzzy, what is that? The loofah squishy thing. That's your proximal convoluted tubule with a microvilli brush border. And then the clean looking distal convoluted tubule, right? No fuzzy border there. And then I had trouble with this one, the thin or thick loop of Henle. It's, I don't know, kind of like a hallway, like a steam room, I guess. That I had trouble with that one. But then the collecting duct, nice cuboidal tiled corridor, like you're walking out of your spa, I guess. I don't know. If you have a better one, please let me know. I would love to hear it and include it. All right, until next time.